All right, welcome to Unit 3. Unit 3 is all about making babies and kind of the rest of the units as well. So, when we're talking about our characteristics of life, one of the big ones is reproduction and development. So reproduction, growth, and development are all categorized under cellular reproduction. It doesn't matter if you're making a new version of a human being, you have a baby, that whole process, or whether you're copying bacteria and letting bacteria grow in a petri dish. All of these things support the cell theory where all cells come from previous cells. And so we're going to be talking about asexual and sexual reproduction as well as genetics in this unit. Okay. For chapter 8, here are your chapter objectives. Again, make sure that you can always answer these, think of them as questions, and you can always find these and read about them in the textbook as well. These are directly from the textbook. Okay. So let's get into it. What is the basis of cellular reproduction? Well, cellular reproduction is how organisms grow and repair. If you've ever gotten a cut, you don't still have that cut because your cells repaired themselves. They replaced the ones that were damaged or broken or lost. And so we know that as such, all cells come from other cells. Okay, and this is really important for both the growth of individuals and new individuals being produced. If this didn't happen, then life would not exist. Okay, this is kind of a big one. But in order for this to occur, of course, we need energy, and so we need to know about cellular respiration and photosynthesis. There are two kinds of cellular reproduction. You have asexual or sexual reproduction. When you have the word or the letter A or an as a prefix to a word, it means without. So asexual reproduction is without sex, and this is generally considered mitosis, but it can be something like when amoebas reproduce, that's called binary fission, and they kind of just split in two. Okay. Sexual reproduction is most often meiosis. There's a lot of different sexual reproductions out there. If you get into a Bio 112 or other classes, you'll find out that that's more than just meiosis, but we're going to learn about the basics of sexual reproduction through meiosis and asexual reproduction through mitosis. Okay. Again, these processes are really important. You need to be able to repair damaged cells. We need to be able to grow. We need to be able to duplicate the DNA and organelles and do so accurately and at appropriate times. As such, cellular reproduction is incredibly heavily regulated. Your cells don't just divide willy-nilly, and when they do start dividing willy-nilly, we will talk about that, but that is generally considered cancer or cells dividing outside of control. So, let's look at mitosis and cytokinesis. So mitosis is the process of asexual reproduction where cells are splitting in two, okay? Cells are making copies of their DNA and are making two cells. We're dividing into two nuclei, which is actually mitosis. Mitosis is concerned about the division of the nuclear contents. And then cytokinesis, cyto meaning cell and kinesis meaning to move. So cytokinesis is cell division, breaking into two parts. So cytokinesis is actually responsible for splitting all of the mitochondria, all of the Golgi apparatus, right? All of those organelles, as well as generating two new plasma membranes or generating, in the case of plant cells, a new cell wall. So when we look at a slide of mitosis, okay, not all cells are rapidly growing all of the time, but if you look at a root tip cell, you will see cells that are consistently in the point of dividing and growing. We know that cells are dividing when we can see the chromosomes clearly rather than just seeing a nucleus. So this image here is from Google, okay? There's tons of cell slide pictures out there. And this particular one I loved because it showed this division of chromosomes into different parts. We're going to see these stages a little bit later on and learn them better. But it also shows just kind of what the cell looks like when it's not actively dividing. You will see the nucleus, and that's about it. But once that nuclear envelope dissolves, we can actually see the chromosomes. And so we're going to talk about the stages that occur here. Okay? Mitosis and cytokinesis are controlled and are part of the cell cycle. The cell cycle includes also a stage called interphase, which is when the cell is doing its job. Okay, a lot of times you'll hear that interphase is when a cell rests. That's not accurate. Cells in interphase are doing whatever their job is. So if it's a muscle cell, it is responsible for moving around, moving your body. If it's a liver cell, it's doing its job of detoxing. It's got probably a lot of smoothie R depending on what you're exposing your liver to, right? If it's a kidney cell, it's cycling your blood and pulling out anything that's a waste product and producing urine. So these cells are constantly doing their job. They are also often going through 3 phases within interphase, G1, S, and G2. Okay? During G1, a phase it's right after cytokinesis. The cell is going to be much smaller, and so it's going to grow to what is considered the normal size adult cell. If I take a piece of Play-Doh and I split it into 2, I have 2 smaller parts of Play-Doh. They can grow as a cell into a larger size. This is also where we're going to see organelles doubling, because if I halved the number of chromosomes or organelles between 2 cells, I'm going to have half the number of organelles, and so those organelles will also duplicate themselves, okay? After G1, the cell might get the signal to go into the S phase of interphase. The S phase is where DNA replication occurs, okay? If I'm going to make a copy of myself, I need that DNA to be replicated before I'm ready to split it, okay? It's like preparing to cook a meal. If you go to cook a meal and you realize that you're out of a key ingredient, you have to stop what you're doing and go to the store and get it. But if you check the recipe and you have all the ingredients ahead of time, you can just go ahead. The S phase occurs between G1 and G2 so that the DNA can be replicated. Sorry, that one was hard for me. The DNA can be replicated and also checked. It can also be proofread, okay? After the S phase, the cell will go into the G2 phase where growth occurs. Again, the cell will get a little larger as it prepares to divide, okay? If the cell then receives a signal, it will go through mitosis and cytokinesis. Mitosis is made of prophase, metaphase, anaphase, and telophase. You've probably heard those words before. This is usually taught at some point in elementary science. And then you'll see cytokinesis following. Okay, this is a normal cycle. Now, every cell goes through its phase of interphase and division at different points in time. Skin cells replicate about once a week, once every 10 days. Stomach cells are roughly every 3 days because your stomach is being exposed to acid and so it's being broken down more. Those sorts of things. Timing is different depending on what the cell is doing as well. And you might have noticed that there is something up here called G0. There is a G0 phase, okay? G0 is for cells that aren't going to go through any sort of division. And this is generally things like your eye cells. Your eyes get worse as you get older. They're not actually replacing themselves as readily. They're not going through that process of repair as easily. That's why eye cells don't readily divide, okay? Other things are things like your heart. Once you're a full-grown adult, generally your heart does not divide anymore, so heart damage tends to be relatively permanent, right? If you have a heart attack, you have scar damage from that. It's because those cells aren't going through ready division. So G0 is cells that are they're at their growth point and they're doing their job and they're not targeted for division. Okay, neurons also fall in here as well. So like nerve damage tends to be permanent because those cells don't readily divide. Okay, this cell cycle again is heavily regulated and is normal process. It's going to go through that on whatever that cell's cycle is. Okay, let's talk about what happens during the S phase. So during the S phase of the cell cycle, chromosomes are duplicated. Okay, this goes through DNA replication, which we'll talk in more detail about in chapter 11. But your DNA is replicated into a duplicated chromosome. The duplicated chromosome is made up of sister chromatids that are identical. The sister chromatids are held in the center by a centromere. This is why our chromosomes tend to have an X shape when we see them drawn out in like a cartoon sort of format. Now, a chromosome consists of one chromatid. Once it is replicated, it is still a single chromosome, but we call it duplicated chromosome, and it has 2 chromatids. After mitosis, those chromosomes are separated, those chromatids are pulled apart, and we get 2 new chromosomes, but those chromosomes are identical. Okay, I want my skin cells to have the same DNA as the previous skin cells. I want my DNA to stay consistent. I don't want mutations. I don't want damage. And so I want these cells to have the same genetic information. These are called sister chromatids because they're identical to one another. They have the same information. They are literally a carbon copy of one another, but then they are divided into the two new cells. Okay, so chromosome of one chromatid, a duplicated chromosome has two sister chromatids held by a centromere, and then your mitosis will divide those sister chromatids into two separate chromosomes. Okay, vocab is very, very important in this unit. We are going to talk about sister chromatids and things consistently. You need to know these vocab words. I strongly recommend flashcards for this unit. There is a vocab sheet for this unit as well in Canvas. So do make sure that you are looking at that and working on this vocab. Okay? All right. So how do these chromosomes get separated, right? How do these duplicated chromosomes get pulled apart properly into new nuclei? Well, this is where mitosis occurs, okay? Mitosis is divided into 4 stages, but these 4 stages roll into one another, and it is pretty continual. Prophase, metaphase, anaphase, and telophase are all part of mitosis. We call that PMAT, okay? Prophase is when the chromosomes are visible and they're condensed in pairs. Metaphase is where we see the chromosomes line up in the middle. Anaphase separates that centromere and pulls my sister chromatids to opposite sides. And then telophase reforms the nuclear envelope. Okay, cytokinesis often occurs at the same time as telophase, but they are 2 separate things. Okay, remember that mitosis includes PMAT, and then cytokinesis is separate. It's its own step. It's its own process in the cell cycle. Okay, so let's take a look at these in images and just a little more detail here. Okay, so in interphase, the cell is doing its normal processes. DNA is replicated to the S phase. It gets all the good signals. It's a skin cell. It's ready to divide. It's doing its thing, okay? And that cell is going to get a signal to move into prophase. During prophase, I will start to see my nuclear envelope break down. So that nuclear envelope, remember, is keeping your DNA separate from the rest of the cell. It's keeping it protected. But if I want to move the DNA to opposite ends of the cell, I have to break down the barrier that's stopping it from moving. So the nuclear envelope is fragmented by enzymes. It's broken down. And then those sister chromatids are still held together by the centromere. Okay, this is going to be just kind of loose in the cell. And if I go back here, some of these cells, the nucleus is starting to look really kind of large. So this cell that's like 3rd row down, 2-3 cells over from the left is definitely in prophase. We can start to see the nuclear material. We can start to see those chromosomes. The nuclear envelope is starting to break down. The nucleus looks very large and kind of loose. That's what we would consider prophase. Okay, so during prophase, we're going to also see spindle fibers start to attach to the centromere, and those spindle fibers are responsible for moving those chromosomes around in the cell. Okay, after prophase, the cell will move into metaphase. At metaphase in mitosis, I will see a single line of duplicated chromosomes, and they are going to be at the spindle equator or somewhere in the middle of the cell. Now, cells are 3-dimensional, and so if I see a slide of a cell, that metaphase might be somewhere generally in there. And so it might not always look like a nice clean line of X's like we see in these beautiful pictures that they drew and took here, because if I'm putting it in a textbook, I'm picking the best picture, right? So let's come back to our picture here. I don't see any really readily in metaphase, and that's okay because this is just a snapshot of cells in their cell division cycle. And so I might not see them readily in each stage if I'm looking at a living specimen, okay? Anaphase is the next phase, and that's going to be where I see those sister chromatids pulled apart. Those little centromeres are still held by the spindle fibers, but I'm going to separate those chromatids. Definitely in this slide image here, we have this big cell, 2 down from our cell in prophase. Absolutely in anaphase, we can see those centromeres pulling apart. You can see the chromatids kind of form that kind of L shape. To me, they always looked like spider legs, and spiders are arachnids, and arachnids and anaphase made sense. I don't know. People say they are pulled apart or pulled away, and I go, yeah, that makes sense. Okay. So my chromatids are separated. They are once more chromosomes, and they are going to be pulled to opposite ends by that spindle fiber. Okay. After anaphase, we will say telophase and cytokinesis. Telophase will be the reforming of the nuclear envelope, and then cytokinesis will be the division of the rest of the cell. Back here, actually the cell next to anaphase is showing telophase. So what we see is these chromatids, which are now chromosomes, are starting to condense. The nuclear envelope is starting to reform. And because this slide is of a plant cell, there's actually something called a cell plate forming. It's really faint, but there's a little cell plate forming in the middle. And that is going to form a new cell wall. In animal cells, we don't have a cell plate. We have a cleavage furrow, which is what this image is showing, where it kind of just pinches that ball of cell into 2. In any case, we can absolutely see cells doing this under various slide images. There is the virtual microscope through your lab, as well as through the NC State virtual microscope. You can absolutely go. I strongly recommend, or even just Google mitosis slides. There are tons of images out there. See if you can identify each of these stages in the cells that are doing them. And you'll be able to see that not everything is clean cut and pretty, but if you practice identifying the cells, that'll be really useful because you will be asked to identify cells on the exam. Okay, so once we know that we have metaphase, prophase, metaphase, anaphase, and telophase, we can talk more about cytokinesis. Okay. Cytokinesis is going to be where, again, in plant cells, we see a cell plate, and in animal cells, we see something called a cleavage furrow. This makes sense, though, if we remember the difference between plant cells and animal cells. Plant cells have that cellulose cell wall. And if I were to take a box and try to split it into 2, I couldn't do it by pinching the sides, right? If I try to take a shoebox and pinch the sides, I wouldn't be able to make 2 boxes. But if I want to make 2 separate spaces in that shoebox, I can put a divider in the middle. Your cell plate is basically a divider. This is going to be a lot of vesicles full of cellulose that are going to then separate this cell into 2. So this cell has the membranes are reformed around the nuclei. I have 2 new nuclei, and I am forming the cell plate. Okay. In animal cells, it's much easier. There's no cell wall there, so there's nothing hard keeping it in the way. And this we could just pinch into the middle and pull apart into 2, and that is what we see happen. Okay, so cytokinesis is very different between plants and animal cells. Now, before we go to the check your knowledge for this part, I do want to show you my little hand thing, okay? And this is kind of weird, but I had a teacher show me this a long time ago, and it's something that I like to go through to help people remember mitosis. And honestly, I see people do it during the test, so it works. If we go, your cells, okay, if you have your hands in a ball, this is interphase. In interphase, the nucleus, and we're only talking about the nucleus, okay? The nucleus is going to be condensed, surrounded by that nuclear envelope. You can't see any of the chromosomes. It's just a circle in the middle of the cell, okay? During prophase, I start to see space forming. My nuclear envelope dissolves, and so I can start to see my individual chromosomes. Okay, during metaphase, my chromosomes align in the middle, and so I have that nice line up of those X's, right, those duplicated chromosomes. During anaphase, the centromere separates, and my sister chromatids pull apart, okay, my little spider fingers, okay? And then during telophase, my nuclear envelope reforms, and I have 2 new nuclei. I can't show cytokinesis with this because cytokinesis is outside of mitosis. So again, interphase, prophase, metaphase, anaphase, telophase, okay? People tend to find that helpful. I think it's helpful, so please use it as it helps. All right, let's look at your check your understanding. Make sure that you can answer these. Make sure you can draw and label the steps of mitosis. You absolutely can draw circles and X's, okay? I think most kids learn to do that very early on, so I hope you can do that. Or you can also go on, like I said, and Google some of those cells and see if you can identify slides, okay? Once you've done this, I'll see you in the next section. Okay, let's talk about a caveat. So, cells do generally end with cytokinesis, okay? But not all cells that go through mitosis go through cytokinesis. One of my favorite examples is slime molds. I'm going to talk about this in a second. Love me some slime molds. But let's first talk about muscle cells, because muscle cells make sense, okay? Muscle cells are long fibers. And so if I'm talking about like my bicep, okay? My bicep is going to attach up here and wrong way, down here, okay? So my bicep is a long bunch of muscle fibers that are attaching at my elbow and at my shoulder, okay? I don't necessarily want these cells to go through the process of mitosis and have tons of little divisions in here. Because if I'm trying to break something, it's very easy to do if it's perforated, like a perforated page, right? Think about ripping a page out of a notebook. If it's perforated, it's very easy to do. You can follow that perforation, okay? That would be what your cells would do if your muscle cells went through cytokinesis. Instead, muscle cells still go through mitosis because we do need multiple nuclei to send the information to make the proteins to do the things, and muscle cells use a lot of proteins, okay? But I'm going to see a nice long muscle fiber that is going to allow me to move my skeleton more effectively and not have concern for tears across it. And so some of those cells can become multinucleated. Okay, multinucleated cells in vertebrate animals are definitely your muscle cells, and I have an image of that here where we see these long fibers. These are all one cell, but those cells are going to have multiple nuclei in there, providing directions for the area directly around them. And again, that makes sense, right? I don't want my muscle to be perforated. I want it to be able to load, and so I'm going to see that muscle cell being a nice long fiber that's not broken. Slime molds are my personal favorite example of this. So if you want like a little extra credit question here maybe, right? Slime molds are the coolest. So slime molds are a protist. They are not related to fungi. They are actually a protist. They are their own thing. They're kind of like amoebas. And if you find a slime mold, which this one here is called the many-headed slime mold, it's very, very common here in North Carolina. It grows into its food, and it is one large organism that is just multinucleated. It is full of cells or nuclei, but it is all one cell because how it moves is by pushing its cytoplasm against the membrane and then growing into that space. Go watch some videos on it. There are videos out there of slime molds moving. It is fascinating. Some people get creeped out. I think it's fascinating. But they couldn't move effectively if they had a bunch of cell membranes stopping the cytoplasm from being able to push into those outer membranes. When we talk about cells and we're talking about the basics, yes, cytokinesis follows mitosis, PMAT, right? Prophase, metaphase, anaphase, telophase. But if the cell's function is to be different than that, we might not see those same steps occur. So which is why cytokinesis is considered separate from mitosis. Mitosis is nuclear division, which we can see with and without the presence of cytokinesis. Okay? Let's talk about what happens when the cell cycle is out of control. So the cell cycle has lots of checkpoints. I'm going to talk about 3 main ones, but keep in mind that there are tons of checkpoints here. I took a biology of cancer class one semester, and all we talked about were cell checkpoints and how cells overcame them. What ends up happening is cells are going to get checked in G1, G2, and M. Okay. In the G1 checkpoint, the cells are checked to make sure that their DNA is okay. I don't want to go through the process of replicating DNA if my DNA is damaged, if it has mutations, if it's not able to be repaired. I probably don't want that cell to make copies of itself. And so the cell will get checked. If it fails the check, it goes through something called apoptosis. Apoptosis is programmed cell death. And this is kind of what you'd see happen after maybe like a sunburn, okay? Your sunburn is actually going to be damage to the DNA that cannot be repaired. When that cell's DNA is checked and it cannot be repaired, the cell is told to kill itself, right? It's told to go through apoptosis. It said, hey, you are not functional. You cannot reproduce. You need to die. And so that skin cell dies, and then you get that flaky skin after a sunburn. That's what's happening there, okay? Same thing if we go through the S phase and the G2 phase and we end up with, again, mutations, damage, errors, something isn't good, that cell is going to get checked again before it goes into mitosis actively. And again, if that doesn't work, maybe the replication didn't happen. It can't divide itself with having identical daughter cells at the end, and so it's going to be told no go, can't pass go, go through apoptosis, okay? Last but not least, there is a checkpoint in the metaphase of mitosis, because if the chromosomes don't properly align in that middle, then they're probably not going to be pulling apart appropriately into the two new cells. And remember, my goal of mitosis is asexual reproduction. I am trying to make an identical cell from the cell that came before it. So I don't want to have one cell that suddenly has too many chromosomes and one cell that has too few. I need that to all be identical and moving appropriately. So these checkpoints are in place to make sure that when cells are being duplicated, when they are going through that replication process, they are making identical versions of themselves so that those cells are going to do their jobs appropriately, okay? Apoptosis can happen at any point here or at any other point during the cell cycle, especially if like the cell gets infected by a virus. It might be told to go through apoptosis to prevent other cells from being infected. So we'll see that apoptosis is very, very normal. This is part of programmed cell death. It helps keep the number of cells at an appropriate level. I said before that your skin kind of is replicated every 7 days. You don't keep developing skin. Some of that skin wears off, and that can be part of apoptosis as well. This can also be a very normal part of growth and development. So if you've ever grown tadpoles and had them grow into frogs, frogs do have apoptosis of the tadpole tail, and those cell goodies, the stuff that's in that cell, is then used by the growing organism to develop the legs, because it's materials, it's nutrients, it's energy, okay? In addition, when you are a developing fetus, we as humans have webbing between our fingers and our toes. That webbing is broken down during development through a process called apoptosis, okay? The best part about apoptosis is that it contains a scientific word, bleb. Bleb is a scientific word. It's the best. So basically the cell will get a signal that says, hey, something is wrong. It will start sending out those lysosomes, start producing these enzymes that causes the plasma membrane to blister or form blebs. The cell is then broken down into fragments, and then your immune system will come in and break them down further and utilize those parts again. Isn't that cool? Okay. So that's the normal process. But if a cell bypasses any of the checkpoints and starts growing out of control and it has acquired mutations, okay, then we can see something become cancer. Now, not every cancer is cancer with the big C, okay? Sometimes it's just the cell is developing abnormally, but you're not going to say it's cancer. In fact, a lot of things have changed where we don't say precancerous cells anymore. We just say like abnormal cells, especially with more scanning and better medical treatments that we have. A cell with a mutation might be abnormal, but it's probably not precancerous yet. And so we're, avoiding that. But let's say that this is your skin cells, okay? Skin is exceptionally prone to cancer when you get UV damage, because UV damage causes mutations in the DNA. If that mutation occurs, there's a good chance that we're going to see more than one mutation, and that may result in those cells dividing out of control. Those cells may now be dividing every 24 hours instead of every 10 days. Obviously, that's going to be very, very different as far as its normal cell cycle, and it has other ways to overcome that. Now remember, I took an entire semester-long class on the development of cancer, on the biology of cancer, and one of the things that I learned from that the most is that, one, every person's cancer is unique, and so we will never have a unified cure for cancer. Okay. And two, there are so many steps that your cells have in place to prevent this from happening. But it can happen, and, prevention and screening are very important. Okay. if these cells start looking abnormal, now you go in and you have a mole that's looking a little suspicious. So you go see your dermatologist. They're not going to say precancerous. They just might say abnormal, and they might remove it just to prevent potential issues. But if you notice it and you go in, cool. If you don't, and that cell starts to acquire mutations that allow it to form a tumor, where the cancer is growing further and bigger and larger and faster in one location, we do see it start to form a tumor, okay? Now, again, once a mutation is found, it can start to accumulate more. And so eventually we might see mutations that start to allow the cells to invade underlying tissue. Once those cells can invade underlying tissues, they also often go through something called angiogenesis, which is producing blood vessels that flow to them. Because again, these cells still need oxygen, they still need glucose, they still need all of that in order to do ATP to go through this whole process. And so those cancer cells might start invading in order to find those resources. You might see blood vessels flowing in there, and then cancer cells have the ability to often break off because they are not tightly grouped together, and they can then become metastatic or metastasize and become malignant and move throughout the body, okay? These cells, again, they don't undergo apoptosis. They have a way of saying these are not the cells you're looking for. They have a way of avoiding your immune system. They often have abnormal nuclei. Some cells have like multiple nuclei, and we see a lot of looseness. So the cancer cells in this image are stained orange, and these cells should be tightly clumped, like your skin cells. They should be tightly grouped together through what's called an extracellular matrix. Cancer cells tend to be looser and not really having that close association, and that's one of the reasons they can break off and become invasive and spread, as well as then having those cells being able to do angiogenesis and generating new blood vessels. Again, that can be a problem. Now, proper screening, right? Tumor removal, chemical treatment, very easy way to treat cancers. If we catch things early enough, a lot of cancers are very treatable now, and that's why regular screenings are very important. Now, the way that we generally treat cancer is radiation therapy and chemotherapy. And what's really interesting about these is that it damages the DNA or some aspect of mitosis, which can lead to side effects. But what it's trying to do is interrupt that cell cycle. It's trying to interrupt that growth process, okay? What happens, and what's interesting, is if a cancer patient is undergoing extreme chemotherapy, they may have issues with their skin becoming really thin and fragile. They may lose their hair. They might have stomach issues. And that's because those cells are all rapidly dividing cells. Chemotherapy is generally meant to target rapidly dividing cells because that is one of the key traits of cancer. But because cancer is your cells, we can't say only target some of these. It's just kind of a blanket treatment, which is why it's great. It can be really useful, and it's very, very effective for a lot of people, but it can be so damaging to other parts of the body because you can't say damage only these rapidly dividing cells. Hair cells, skin cells, stomach cells are all considered rapidly dividing cells because they do divide, every couple of days rather than every 2 months, 3 months, once a year, whatever it may be. Okay. Other cancer treatment options that are out there, and some of the ones that are potentially promising, are things like hormone therapy and immunotherapy that are generally built on your own system. Very expensive, not the best way that we have. It probably is one of the best ways that we have to do things, but it's not easily available to everybody. Instead, as I've mentioned before, a lot of what you should do is more cancer prevention, and understanding how cells divide can help you do that. So, some of the things, having protective behaviors, eating plenty of fibers. I've seen a lot of things that say people don't eat enough fiber today, which is true. Sunscreen. Put on sunscreen every day. You should be having sunscreen just in your lotions, right? Heavy drinkers. There are genetic factors that play a role, right? Having a protective diet can be really useful. I'm not saying you should avoid McDonald's, but maybe don't eat McDonald's every single day. Okay. Know your own personal risks. If you have family history, make sure that you're doing your regular self-checks, whether that be a breast check, testicular check, whatever. If you have people in your family who've had colon cancer, even if you're younger, it's better to make sure that you're tracking that and having regular preventive colonoscopies, because it can be really dangerous and it can divide very quickly. The same thing with skin cancers. And, just keep in mind that one of the things that I also took away from my biology of cancer class was that we are exposed to more nowadays that do cause cancerous items. Absolutely. We're exposed to more carcinogens now than we used to be. But in addition, people are living longer than we used to. And when your cells are going through division, they do start to just break down and not divide properly. And where people might have died of infections or heart disease or other things earlier in their life, as we're living longer, we are seeing an increased prevalence of cancer because we're living longer, okay? So just keep that in mind. I don't want this to be like a scary lecture. I think it's interesting because mitosis is so important, but what happens when it goes wrong? Because that's always the question, right? But what about when it doesn't work? Okay? All right, make sure you can do your understanding check here. I do strongly recommend watching the Amoeba Sisters video comparing mitosis and meiosis and completing the Venn diagram that's in Canvas. And if you have questions, as always, please let me know. Thank you.